In designing an antibiotic, you wouldn't create a drug that destroyed DNA, for example, because that's something that both humans and bacteria share in common. It would kill the bacteria all right, but it might kill us too. So many antibiotics work by attacking the bacterial cell wall, something bacteria have that we don't. Antifungals can attack the unique cell walls of fungus. Pesticides can work by attacking the special exoskeleton of insects, but fighting cancer is harder, because cancer cells are our own cells. So fighting cancer comes down to trying to find and exploit differences between cancer cells and normal cells. 40 years ago, a landmark paper was published showing for the first time that many human cancers have what's called absolute methionine dependency, meaning you can grow normal cells in a petri dish without giving them the amino acid methionine. Normal cells thrive, but without methionine, cancer cells die. Normal breast cells, for example, grow no matter what, with or without. But here's leukemia cells. They need that extra added methionine to grow, or they just flatline. What does cancer do with the methionine? Tumors generate gaseous sulfur-containing compounds with it that specially trained diagnostic dogs can actually pick up. There are mole-sniffing dogs that can pick out skin cancer. There are breath-sniffing dogs that can pick out people with lung cancer. Pee-sniffing dogs that can diagnose bladder cancer, and yes, you guessed it, fart-sniffing dogs for colorectal cancer. Doctors can now bring their lab to the lab. Gives a whole new meaning to the term PET scan. Anyway, methionine dependency is not just present in cancer cell lines in a petri dish. Fresh tumors taken from patients show that many cancers appear to have biochemical defects that makes them methionine dependent, including some tumors of the colon, breast, ovary, prostate, and skin. Chemo companies are fighting to be the first to come out with methionine-depleting drugs, but since methionine is sourced mainly from food, a better strategy may be to lower methionine levels by lowering methionine intake, eliminating high methionine foods for cancer growth control. Here's the thinking. Look, smoking cessation, consumption of diets rich in plants, and other lifestyle measures can prevent the majority of cancers. Unfortunately, people don't do them, and as a result, each year hundreds of thousands of Americans develop metastatic cancer. Chemotherapy cures only a few types of metastatic cancer. Unfortunately, the vast majority of common metastatic cancers, like breast, prostate, colon, and lung, are lethal. We therefore desperately need novel treatment strategies for metastatic cancer, and dietary methionine restriction may be one such strategy. So, where is methionine found? Particularly chicken and fish. Milk, red meat, and eggs has less, but if you really want to stick with lower methionine foods, fruits, nuts, veggies, grains, and beans. In other words, methionine restriction may be achieved using a predominantly vegan diet. So why isn't every oncologist doing this? Despite many promising preclinical and clinical studies in recent years, dietary methionine restriction and other dietary approaches to cancer treatment have not yet gained widespread clinical application. Most clinicians and investigators are probably unfamiliar with nutritional approaches to cancer. That's an understatement. Many others may consider amino acid restriction as an old idea, since it's been examined for several decades. However, many good ideas remain latent for decades, if not centuries, before they prove valuable in the clinic. With the proper development, dietary methionine restriction, either alone or in combination with other treatments, may prove to have a major impact on patients with cancer. Probably about a third of common cancers can be prevented by eating a healthy plant-based diet, being physically active, and maintaining a healthy weight. One of the ways plants may help is by cutting off the supply lines to cancerous tumors. A tumor cannot grow without a blood supply. Currently, it's believed that a tumor mass cannot exist in a volume greater than about the size of the ball at the tip of a ballpoint pen without a proper blood supply, which indicates that angiogenesis — angio means vessel, so the genesis, the creation of new blood vessels, is critical to tumor growth. Each one of us has cancer cells in us right now. By age 70, microscopic cancers are detected in the thyroid glands of virtually everyone, for example. Most of these tumors never cause problems, never become clinically significant, leading to the concept of cancer without disease as a normal state during aging. Cancer cells are commonly present in the body, but they can't grow into tumors any bigger than that tiny dot size, no more than 10 million cancer cells, before needing to get hooked up to a blood supply. So, tumors diabolically release angiogenic factors, chemicals, that cause new blood vessels to sprout into the tumor. The most important one is called VEGF, vascular endothelial growth factor. But we can suppress VEGF with veggies. Many of the phytonutrients we know and love in tea and spices and fruit and berries and broccoli and beans can block cancer's stimulation of new blood vessels. They're ideal for prophylactic long-term use against breast cancer because of their reliability, availability, safety, and affordable price. Dietary agents used to suppress angiogenesis may be an important step in the prevention and treatment of breast cancer, and in fact, all types of tumors. The review concludes that we now have convincing evidence that dietary plant constituents possess the unique ability to affect tumor angiogenesis, which may be deemed advantageous in the prevention and treatment of human breast cancer. Most of these studies have only been done in a petri dish, though. You stimulate human blood vessel cells, and they start forming these tubular structures, trying to make new capillaries to feed the tumor. But if you add plant flavones like apigenin or luteolin, found throughout the plant kingdom, like in citrus, celery, and peppers, you can see they help block the tube formation. Here's the effect of fisetin, a phytonutrient found in strawberries and other fruits and veggies. It just shrinks the beginnings of new blood vessel formation right on down. Where do researchers get their hands on human blood vessels? From human umbilical vein endothelial cells. They get them from discarded umbilical cords, or more controversially, from the eyes of aborted fetuses. But either way, you can stimulate blood vessel formation with the tumor compound VEGF, and then abolish that effect with plant compounds, in this case from purple rice. Therefore, the daily consumption of natural foods containing adequate flavonoids could be beneficial for the prevention of cancer metastasis, or could improve cancer prognosis. Given the power of plants, one might speculate that the foundation of an anti-angiogenic approach to cancer might be a whole food vegan diet.
What I'm going to talk to you about is immune-based therapies. And unlike my colleagues, you'll have to look at me because I have no slides. So I started today talking to you about the transition from the last decade of clinical research and available treatments for kidney cancer to what is really a pivotal time in our kidney cancer history. And as many people think about it, they think that it's something that just began, but in fact it's been decades in the making. So there's 4 areas that I'd like to talk to you about in the next couple of minutes. The first is something called spontaneous regressions. The second is immune-based therapy with drugs like interleukin-2 and what that's taught us. The third is the role of vaccine therapy. And lastly, we're going to talk about a new class of agents that is not yet commercially available, but that is for kidney cancer, but is reaching the clinic hopefully in the very near future. And I can talk to you a little bit about the pluses and minuses of that. So let's start with spontaneous regressions. So it's been known for decades that kidney cancer is one of the few cancers that when you take out the primary cancer, where there's been spread to other areas, that sometimes in the absence of any systemic treatment, those other areas either decrease in size or disappear. That's what's called a spontaneous regression. It occurs very infrequently, but it does occur, well documented in the literature. So one would ask themselves, what's the mechanism for why that occurs? And the mechanism is thought to be that the primary tumor, the kidney cancer in the kidney, makes molecules that suppress the immune system's ability to manage its cancer. And I think that's, even though that's kind of a 1950s phenomenon in our cancer literature, it has really great importance in 2015, 65 years ago. Because we now know that cancers control our immune response very differently than when we get vaccinated. So for example, many of you got a flu shot this year. It's pretty typical. Everybody, you watch the TV and the TV says, get a flu shot. And you would ask yourselves, well, why do I have cancer and my immune system can't manage my cancer when I get a flu shot, and the flu shot can help me mount a response so that I don't suffer from the flu that comes out this year? And the reason for that is that the cancer is very, very smart. It's not that the flu shots aren't smart. And what it does is it produces an environment around the cells that prevent our immune system from seeing it, reaching it, touching it, and destroying it. And that's what's called immune suppression. And it used to be thought that immune suppression in kidney cancer and other cancers was a result of something that happened far away from the cancer cell itself. And the reality is that it's happening right next to the cancer cell, a place called the tumor microenvironment. So the tumor, we always used to think is, well, there's these other cells that are inside, and then there's the cancer cell, and it's the cancer cells that drives the conversation. And we now know that it's not just the cancer cell, but all the other cells that are there. And what we're doing, fast forward, is I'll tell you about how we're manipulating that tumor microenvironment when I talk to you about checkpoint inhibitors. So that was spontaneous regressions. The first hint that kidney cancer might be different. If you said to me, well, Dr. Figlin, that must happen in a lot of cancers. The answer is no, it doesn't. Spontaneous regressions don't happen in very many cancers, and kidney cancer is one of the few and infrequent diseases that it occurs in. So then let's go forward and talk about IL-2 and interferon. So IL-2 is a protein. Some of you may have heard about it, some of you may have received it, some of you may have benefited from it. And it is a drug approved in 1992 that took about 6 years to get approved, so mid to late 80s until 1992. And what IL-2 does is it basically hammers, and I use that word advisedly, it hammers your immune system. It stimulates your immune system in a way that nothing else does. Not vitamins, not certain kinds of green leafy vegetables, not anything that the pharmacist or the TV tells you are immune stimulants, but it's a drug that basically hammers your immune system. And when you hammer the immune system, one of the things that occurs is you create an environment of cells that then seek out and have the capacity to destroy a cancer cell. And in kidney cancer, when you think about kidney cancer now, what is that, 8 and 15, 23 years later from its approval, if you asked an audience, whether it's this audience or any audience, about who are the multiple decade survivors of kidney cancer, it's patients that received IL-2. So Nancy and I have patients that are 20 plus years free of metastatic cancer from IL-2. The problem with IL-2, unfortunately, is that it's very infrequently works, usually less than 10% of the time, and it requires multiple hospitalizations in the intensive care unit where you get very, very sick. And it's not really been adopted by most physicians around the country because, one, they don't have the experience. Two, the frequency of benefit is low, and that's independent of trying to identify the people that are most likely beneficiaries. So one of the questions that I've spent a significant part of my scientific life is to try and identify who most likely benefits. Because, boy, if we could identify the people that would benefit, then we could treat just those and not use that treatment for people unlikely to help. And we have an article that just was published that looks at that. But fast forward 23 years, and although we have tried, we've not been very successful. So the gentleman in the second row asked about chromophobe. We know that it doesn't work in chromophobe. We know that there's certain subpopulations of patients where IL-2 can work, but even so, the success rate is usually less than 1 in 10. But what it did tell us was that the interaction between the immune system and kidney cancer remains a potential for curative therapy. And remember, I used that word earlier. So fast forward to the kind of 2005 to 2012, we're starting to talk about vaccines. And what are vaccines? Well, vaccines are no different than the flu shot that you take. The flu shot is an inactivated flu of certain strains that stimulate your immune system to protect you against a virus. We now have at least 2 large randomized clinical trials in kidney cancer using 2 different vaccine approaches. Vaccine 1 is an off-the-shelf vaccine that's combined with a drug that many of you may have received, Sutent, or a drug like Votrient, where the hope is that we stimulate the immune system against things on the surface of the cancer cell, which when combined with Sutent results in improved long-term control of the cancer. That trial has been completed. We're waiting for the results. Results probably won't be known for another 24 months. The second is a little bit different, and that's a trial where the vaccine is actually generated from the patient's own tumor. This is what's called a personalized autologous vaccine, meaning that it doesn't come off the shelf, it comes from you. And we take that tumor and we process it. We extract what are called the proteins on the surface of that cancer. We grow it in the laboratory. We put it on top of your immune system cells called your dendritic cells, and we give them back to you as a vaccine, much like a flu vaccine, in combination with Sutent. That trial has now about 300 plus patients on it of the 450 that will be required. It's a trial taking place at 160 places around the world, and we'll hopefully have the results of that by 2016, about a year and a half from now. So one can imagine that we are now trying to exploit your immune system on behalf of destroying your cancer. And then the last area, which is incredibly novel, is the realization, and this was actually first described in kidney cancer, mid-2000s, so about 2004, by a group at the Mayo Clinic, Eugene Kwan. And what they described was that there are these molecules on the surface of a kidney cancer cell which make the outcome for that patient less well than if the molecules were not on the surface of a kidney cancer cell. And then fast forward about 8 years, travel from Rochester, Minnesota to Baltimore, Maryland at Johns Hopkins, they realized that these molecules called checkpoint inhibitors are molecules that do the following. The cancer cell tells your immune system through something that it makes to block the immune reaction between your cancer and the cancer cell, between the immune system and the cancer cell. So imagine that this is the cancer cell and this is your immune system. There's something in between that, despite a strong reaction, can never see each other and interact. And that blocker was understood, and as most of what happens in the world of oncology, industry gets involved at some point, and we developed blockers to the blocker. And you do that in the form of an antibody. So what you do is you say to the immune system, I'm going to give you this treatment, and what's going to happen is the immune system that is being blocked by the signal between the cancer cell and the immune system is going to be blocked further by this drug that we're going to give you, a so-called checkpoint inhibitor blocker. Lo and behold, you clone it, you make it, you give it to people, and there are now 2 diseases one and a half, let's say, where this drug has been beneficial, this class of drugs. It's approved in melanoma, and it's soon to be approved in lung cancer. And what's amazing about this approach is that it's evident that diseases that are not thought to be immunologically reactive are now becoming responsive to immune manipulation, lung cancer, for example, where you wouldn't have expected that to occur. So we've participated in the kidney cancer trials. Those trials have accrued, met all of their goals, and we're waiting for those results. And we would expect that kidney cancer will be one of the diseases where these checkpoint inhibitor blockers will be approved, hopefully in 2015, possibly in the first half of 2016. Now, but we've learned a lot, going back to our IL-2 conversation, that only about 1 in 10 benefit, and the same is going to be true for the checkpoint blockers. And that is that despite the fact that it's very exciting, it doesn't appear that everyone benefits from these drugs. And we think that the benefit usually resides in about 20 to 30% of the patients. So again, much like the science that we did over the last couple of decades, it will be, what is it about these people and what is it about this disease that allows these immunologic manipulations to work in some people and not in others? The other challenge is that when you turn on an immune system or you block the things that are blocking the interaction, you then are exposed to other immune reactions that might not be okay. You can develop diseases of the thyroid, of the adrenal gland, of the pituitary, of the gastrointestinal tract, because when you turn on an immune system, you sometimes don't have complete control over the interaction between the immune system and the cancer cell or the immune system and other parts of our body. So while very exciting, also potentially very challenging with respect to how it's given, how it should be combined with other agents, and who are the best people to receive it. So I'll circle back and talk to you about this later when we talk about some of the research that we and others are doing toward the end of today. But I think that my own bias is that the immune approach to kidney cancer, which was quiescent over the last decade, has reblossomed, and that reblossoming of that effort is going to have a profound impact on the kinds of things that we can offer you and patients like you in the future. So I'll take a couple questions because we have a couple minutes, and then we'll move on to some other topics, but we'll have a panel discussion later. Any questions? Perfectly clear? Mr. Cott, you must have a question. Use the microphone.
Is that PD-1 that you were referring to? Is that one of those drugs?
Yes.
Are there other ones like that that might operate differently?
Yeah, so
Where PD-1 was not successful?
yeah, the question that's being asked is, there are 5 companies that are now internationally competing for this space, all of them with a little bit of a different approach. And the question is, if one doesn't work, might the other one work? And we don't have any evidence that that's true yet. There's no data in the literature that would suggest that if A is ineffective, that B can be effective. But we will see those evolve over time, probably within this year. But currently, there's no evidence that that's true.
Kind of following up on the questions about biomarkers from Dr. Luthringer, are there real good tests to indicate which patients at this point are more likely to respond to some of these checkpoint inhibitors, or ways to exclude patients?
Right. So the easy answer to that question is there are no good tests to predict who will be a beneficiary and who will not. Those are tests that are being looked at and evaluated, but there is not a blood test, a tissue test, or some other kind of test that can predict how a person will do, although we look continuously.
We don't talk about immune system in general. We talk about 2 different arms of the immune system. The one that is less relevant for cancer, and the one that is more relevant for cancer. So this is the adaptive immune system. Our immune system needs to remember what a cancer cell is because it's a non-self. So it's not something non-specific. And the key to all this, the key is the interaction of these cells that are called antigen-presenting cells. These are the cells that pick up stuff that is non-foreign from the environment. This can be viruses, this can be bacteria, but yes, it can be cancer cells because cancer cells are non-self cells. So these are sampled from the microenvironment and are being presented in these antigen-presenting cells. And it is this key interaction with these other cells that are called lymphocytes that you saw in the very first slide that is the key to educating the immune system. Okay, so that's a different way of seeing the adaptive immune system. Fancy picture where antigens are being picked up at these antigen-presenting cells. These are called dendritic cells from the Greek word dendros, which means tree. So they have these spiny projections. And it's, again, an uninstructed, naive T cell that is going to come and encounter the antigen-presenting cell, and then it's going to get activated. And either it's going to kill the cells, or some of them are going to be left behind for memory. So that's a very simplistic approach of what is adaptive immunity. And again, the key interaction here is the interaction between the antigen-presenting cell and the lymphocytes. So here's your first kind of complex slide, but the way I'm going to present it is like, it's basically the interaction between the antigen-presenting cell that I showed you before and a lymphocyte, a T cell, is actually so complex that over the last 20 years there have been so many molecules that have been identified to play a role in this interaction. And it's no longer a simple interaction of a signal that means that this antigen-presenting cell is going to present the antigen and, the immune cell is going to get activated. The decision, so it's no longer one what's called first signal. It is a number of other things that the interaction between these 2 cells have to be taken into account. And whatever is in red, it means that these are actually proteins that are going to say, no, I'm not going to get activated. And then those things that are green, these are the cells that say, yes, I'm going to be activated. So the net effect of whether an immune cell is going to actually get activated is the net result of the positive and the negative factors. And it's easy to understand that this looks to me like a complex car with multiple brakes and gas pedals. So that's how complex nature is. And right away, you're beginning to see things that you may have seen already on the internet. You're seeing things like CTLA-4. So there's already a drug against CTLA-4. It has been used for renal cell cancer. It's called Yervoy. It has not been approved yet. Also, you see these other negative immune checkpoint protein that's called PD-1 or PDC-1. That is the target for nivolumab or pembrolizumab that was FDA approved for melanoma 3 months ago. And then whatever you can see in red, it means that there are already drugs in development. So it is no longer just the PD-1 that you see or the CTLA-4. There are a bunch of actually targets that are currently in clinical development. So this is a very hot area of interest. So there's going to be more to come over the next 5 to 7 years. Okay, so this is the gas bladder scenario in normal states. So if you have 2 gas pedals and 1 brake, then it's a go. And that's a nice looking car that actually makes a turn and does not go upside down. And this usually happens when there is an early stages of an infection. Our immune system has to become activated. Very simplistic the way I present, but this is a good way to understand how it goes. And then when the immune system takes care of business in the normal states, then all these, several of these negative immune checkpoint proteins are being upregulated, and then basically you have your car nicely parked, ready to be used for something else. So I want people to understand here that cancer and the immune system, there is constantly an interaction. I remember I was a third-year medical student in Greece when our pathologist said, our body makes approximately 30,000 cancer cells per day. And the only reason that we don't develop cancer is because we have a constantly working immune system. So there is always this tendency, the Darwinian tendency of our bodies to develop mutations, to become something different, to evolve over time. But there is constantly this system that's called immune system that is trying to seal things off. So therefore, any conditions can turn a normal tissue into a cancerous tissue, and by definition, the cancerous tissue is slightly different from the normal tissue, and that is perceived as foreign, as a non-self by the immune system, and in so doing, the immune system is trying to eradicate those cancer clones. And then there is this constant interaction between how a cancer becomes genetically unstable, develops new mutations, okay, becomes more different, and how it actually tries to suppress the immune system, and how this battle between the immune system and cancer is going on and on. And that probably has been going on for years and years to come. This is not an overnight event. And eventually cancers develop because the immune system has failed. So when somebody has cancer, it's basically a failed immune system. So there's not many things to fix it. It's not like if you increase your exercise, or you increase your activity, or you improve your diet, or you get a supplement, it's going to get better. It is already broken, and therefore there have to be ways to restore in the way of drugs. I believe in alternative medicine, but the effects of it are very well unknown in a patient with metastatic cancer. So again, will the future be one size fits all? I just talked to you about one way that the immune system is suppressed, and that is by upregulating these brakes. But there are a number of things that are going on. There are many different ways that a family can become dysfunctional. And I quote one of the very nice phrases that Alexander Tolstoy has wrote in Anna Karenina. There's one way that a family can be functional. People would be happy, and kids love their parents, and vice versa, and everybody's happy. But there are many ways that a family can become dysfunctional. That is how cancer is. There are many different ways that your immune system can become suppressed. So here we go. So if the cancer cell does not upregulate these HLA molecules, the ones that require for the first signal, the immune system is not going to see it. And therefore, that's how cancer cells evade the immune system. That's one way to do it. The other way to do it is to produce all these molecules that basically suppress not only those cells that are supposed to pick up these antigens and instruct those lymphocytes, but suppress pretty much everything. Suppress the antigen-presenting cells, but suppress the immune cells as well. And guess what? Many of these things that are being secreted by the cancer or when the lymphocytes come in contact with the cancer, it can be a toxic effect. So, as this nice review article from Teresa Whiteside, who was my neighbor in neighbor office in Pittsburgh, she says immune cells in the tumor microenvironment are functionally impaired, and I'm adding, and are killed. So your lymphocytes are getting killed by the cancer, by the cancer cells. And you all are patients and you understand how to read a complete blood count test. But what I want to emphasize you here is I'm showing you 8 different patients, CBCs, simple as that. The common with these patients is that all of them have metastatic cancer. And the one thing that makes a big interest is that the fact that all of these lymphocytes are low. So there is a fundamental problem in most of patients, end-stage cancer, that their lymphocytes are low in the blood. Okay, so for people who don't know what is a high-dose bolus IL-2, it has been FDA-approved therapy for 20 years. It's not for everybody. It is a very toxic therapy. Not everybody can do it. It is being given in hospitalizations. This is called a hospitalization is called a cycle. 2 cycles include a course, and we can evaluate things between every 2 courses. Okay, so this is what IL-2 does. So this is our very first patient that was treated at the University of North Carolina with renal cell cancer. So what you can see here is that before he got into the treatment, his lymphocytes were low. And then when he started to get IL-2, his lymphocytes disappeared. And that's what IL-2 does. Makes the lymphocytes disappear from the blood, and they're going to different parts of the body, including the cancer cells, and they're supposed to eradicate the tumor. And what happens is, when the cycle ends, then the patient shows up right before the next dose, and his lymphocytes are almost double right before the second course. So what I want to emphasize, again, do not forget drugs like IL-2. So IL-2 is otherwise called T-cell growth factor. I call it a T-cell steroid. If patients' way of immune suppression is actually they just don't make a lot of immune cells, IL-2 could be a very good drug to give. So the same thing now, but now I have this thing there, which means that the cancer cell can actually immunosuppress because it can also develop properties of antigen-presenting function, and then all of a sudden you see that the lymphocyte can take wrong instructions from the cancer cell. As you can see, there are more breaks that the cancer cell can induce as opposed to gas pedals. And this is now the scenarios when somebody has cancer. Okay, so you may have advanced cancer, and your first signal is functioning, and you have 2 brakes and 1 gas pedal, and therefore you're looking like a Formula One car that is on high brakes. But if, for example, your immune system, the cancer does not have expressing those things that makes the cancer cell visible, then you're like a car that is driving in the middle of the night without knowing where things are. And you can have the third situation of immunosuppression where things are functional, but the cells are dying because the cancer is suppressing them. And this is a car on fire. So what the immune checkpoint inhibitors in renal cell cancer, and that seems to happen in 50% of patients with renal cell carcinoma, is that you may actually be able, with one of these drugs, to eradicate one of the 2 gas pedals, and that could be sufficient for the immune system to get activated. And sometimes, if you use 2 of them at the same time, okay, and this is the PD-1 plus the ipilimumab, then this immune system is really revved up to the point of going up in the air. So this is a most updated list of clinical trials using these immune checkpoint inhibitors in various cancers. And I want to show you the renal cell cancer in relation to other cancers. So what I want to show you is that if you use these 2 drugs, the ipilimumab and the nivolumab, in various doses, okay, in combination, you can rev up the immune system to such an extent that you can see response rates of as high as 44, okay? Now, if you use only nivolumab in renal cell cancer, then the responses are less, but nevertheless, much better for responses that you get in other cancers like lung cancer, ovarian cancer. So what I want to emphasize is that different cancers would respond to these drugs in different percentages, and it seems that the renal cell cancer behaves in terms of response rate similar to melanoma. So again, see where the ipilimumab and nivolumab was given in patients with lung cancer, the response rate was only 16%. So what are the benefits and challenges of this new class of immunotherapy? So definitely the way we see that in melanoma, when ipilimumab was approved in 2010, we were able to give immunotherapies in a much larger number of patients. We were giving ipilimumab in a 90-year-old. We were giving ipilimumab in a 70-year-old with congestive heart failure. We were giving ipilimumab in someone with coronary artery disease. IL-2 cannot be given in those patients. So the application of immunotherapies will be much larger in a larger group of people, the same way we saw that in melanoma. What else would be convenient? It's going to be an outpatient therapy. You don't have to be admitted for this horrible 5-day course of high-dose bolus IL-2. And in fact, if you come to the MICU, the Medicine Intensive Care Unit at UNC, you're not even going to have a bathroom. So it is definitely less toxic, okay? So that's no questions asked. But again, we have to see the less toxicity in a different perspective. The benefit of IL-2 is that you will do it for 2 months or 4 months or 6 months, and then you're going to be done. With these kinds of immune checkpoint therapies, potentially the side effects can be longer lasting, in particular if you use the combination of ipilimumab and nivolumab. Will they be more effective? So in our mind, we have linked response rates that I just showed you with clinical benefit, which is progression-free or overall survival. The trials are too early right now to say whether this high response rate would actually lead to longer survival, but it's very safe to think this way. But unless we do follow these trials over a longer period of time, we will not be able to know the long-term benefit. And the challenge is we're talking about drugs that are, it's $20,000 an infusion here. So we should do a better job, we as doctors, to really refine which patients actually are candidate for. And I think I predicted that this combination of drugs can be effective in up to 50% of patients, but definitely not 100% of patients. And for how long we're going to give these drugs? So right now, the drugs, at least the approval of the pembrolizumab for melanoma is basically indefinite. So as long as you have cancer and you respond to that, then you're going to be getting pembrolizumab forever. Is that the right thing to do? I mean, this provides a huge burden to the insurance companies and the healthcare system. And again, as I said, does response rate correlate with long-term benefit, and what's the cost? And how to combine these drugs with other drugs. And I intentionally did not do that because I would wait for you to make your questions in the panel discussion. So my summary and conclusions is that future is for some, but definitely more than the lucky IL-2 candidates. So you shouldn't be privileged because most of you will be candidates for both of these drugs, the ipilimumab, the pembrolizumab alone or in combination with ipilimumab. But not one size fits all. I think I showed you that there are many ways that cancer, i.e. the family, can become dysfunctional, and this is one of them. And do not forget that there are also good old drugs. So tyrosine kinase inhibitors, some of them, I've seen that they have response rates 20, 30, 40%, and people have been on that for years and years to come. And there are also high-dose bolus IL-2 that you should not forget. But if the patient is not, is in an advanced stage of disease, you just cannot beat up a dead horse with any immunotherapy. in which case you have to use other types of treatments. So these treatments are not for everybody. So I thank these 6-7 patients and their families who have trusted us to give them high-dose bolus IL-2 in the hospital, and I'm sure that they are eager to see something less toxic than IL-2, and the GU program that refers patients to us for this. Thank you.
Okay, well, thank you very much for inviting me. I want to thank the organizers here at NYU and the Kidney Cancer Association. And I would like to just say that some of the things that Dr. Alter mentioned in terms of counseling patients are really the bottom line, which is what I always say to people is the name of the game is staying in the game. And Sarah and Paula are examples of that. And some of you are, I know. And what we've learned over the years is that we can find new treatments and new pathways, and that's what we're also trying to do with some of the data that you've seen. And I'm going to just talk a little bit about, which way am I going? Side, oh, those are my disclosures. Okay, there we go. Why immunotherapy? So we're going to talk a bit about why immunotherapy is even a consideration for kidney cancer, talk about the older immunotherapies, although Dr. Alter gave you a good introduction to that, and then some of the clinical trials that are coming up in the near future. So why do we think about immunotherapy? Well, that's always been the holy grail of cancer treatment, get the body to get rid of this foreign whatever. But the problem is that, cancer is us. It's a little bit different. It's a little bit manipulated or mutated, but it comes from our own body. So we have to try to figure out what about the tumor is different and how we can go after it. And that's been the goal of immune therapy. But the reason kidney cancer has always been a target for that is because there's often a long hiatus. People will have a kidney removed and 5 or 10 years later, something will show up and you say, what the heck? It's doing it again. So what kept it in check for all of that period of time? We also know, speaking of doing surgery in the setting of metastatic disease, it's rare, but we've all seen it, that somebody may come to you with small lung nodules and a kidney tumor, and the kidney tumor is removed, and then you do another scan before starting treatment, and lo and behold, all of the lung nodules are gone. And what did that? What was that about the primary tumor that was driving the metastatic disease? Was it a growth factor produced by the tumor, or did the immune system go after those tumors once there wasn't such a bulky burden of cancer? We don't know the answers to those questions, but those are the intriguing observations that have led us to encourage immunotherapy as a potential therapy for kidney cancer. And as Dr. Alter showed you in his remarks, there is demonstrated responsiveness to interferon, which I'm not going to spend a lot of time on, so I'm grateful that he did, and interleukin-2, both in the laboratory, which is in vitro, and in animal models and in people. So who are candidates for immunotherapy? For over the years using interferon and interleukin-2, we've said, we've kind of learned that, as Sarah even commented on, and Paula, clear cell seems to be the type of renal cell that responds the best to interleukin-2 and interferon. That's what I say so far. But the checkpoint inhibitors that were briefly mentioned may in fact have a broader spectrum. So think about it this way. Interleukin-2 and interferon are substances your body makes normally in small amounts, and what we're doing is giving you supraphysiologic amounts, and we're stepping on the gas. We're pushing the immune system to go after the tumor cells. We're getting cells that are in your body that normally have a very short activation period to stay activated and go after those tumors. Then we're taking these newer drugs, the checkpoint inhibitors, they take the brake off. So what we're doing is stepping on the gas and taking the brake off, and Paula said that's how she drives anyway, with both feet. I don't know, guys. But at any rate, that's the concept of looking at those two things together. And there are the beginnings of some combined clinical trials coming up. So why is IL-2 still in the picture? Because of what Dr. Alter mentioned, there are durable complete responders. There are people that have been treated 20 years ago, had a complete response, and have no disease, residual disease in 20 years. There are people who have gotten that disease, had a shrinkage, and then it stayed stable. We don't even know if what we see on the CAT scan is active disease. that happens as well. So it's that hope for that complete or durable response that keeps this drug out there and keeps people going through a fairly rigorous treatment, but a short-lived treatment. So what we learned over time was that with clear cell, overall, there's about a 20% response rate. The original papers said 15, now it's up to 20. Most recent study we did was 25%. Clear cell with favorable features, which means a very pure clear cell histology, which, by the way, reflects a molecular profile, which will be talked about in the subsequent presentation. The response rate went up to 40%. Now, this is retrospective data. This is looking back. So it may or may not be quite these numbers, but we do know that the histologic subtype and or its molecular profile make a difference in responsiveness, at least to interleukin-2 immunotherapy. Okay, so as was mentioned, it's a growth factor for T cells, which are the killer cells that are activated to go after tumors. It expands these populations of killer cells, and then we keep stimulating them over the course of the treatment for those 5 days so that they go to the tumor, they infiltrate the tumor, and even after we stop the treatment, those cells are working in the body because they're alive. They're part of the body's activity. There also are secondary cytokines that are produced that are inflammatory, that are interleukin-1, tumor necrosis factor, gamma interferon. We still don't really know how well they play into what's happening with the treatment. But in the laboratory, more than 90% of tumor cells are killed. In humans, when we give the drug, it's about 20 to 30% of the patients have major responses. So we really are still trying to figure out who and why. Okay, so this was the original data that led to the FDA approval more than 20 years ago. And at that time, there was a 15% response rate, but the key was the durable responses, and I'll show you that data. This was a study done at the National Cancer Institute by Jim Yang and his colleagues years ago, and it used 3 different doses of interleukin-2. It used high dose, which is the inpatient every 8-hour treatment. It used a log less of the drug, which we call moderate dose. So it's, again, that every 8 hours, again, 5 days on, 9 days off, 5 days on. And then a sub-Q regimen that's been used as well. And what we saw was that with the 2 lower doses, if you look at the bottom column, that's the 3-arm study. The 2 lower doses, the response rate was about half of the high dose. And that told us that the high dose was really the most active. But the other part to say that we actually, with our cytokine working group, did a study of the lower dose intravenous. And for people that have medical problems like heart disease or pulmonary disease or kidney problems, they can tolerate that lower dose. And in fact, they can get responses. So there's a trade-off in terms of the overall response rate, but it's not negligible. And for some people, it's the answer. And here, what you see here, those numbers across the top with all the pluses, that's how long those responses have lasted. And this is an old slide. But the point is there are people that are out more than a decade with responses that have lasted after that one cycle of treatment. So that's the carrot for immunotherapy. Okay, so we did a trial called the SELECT trial where we were trying to figure out who are the responders. We still haven't teased out all of the data, but clearly we were looking at histology, the type of kidney cancer, we were looking at the immune system, and we were looking at markers, that B7H1, which is a marker of aggressiveness, but in fact it's also a target now that anti-PD-1 antibodies are using. And we again saw, by this clinical selection, we got a response rate of 30%. So we doubled what was in the package insert because that data included people like Paula with the papillary and some of the other subtypes. So the summary of IL-2 status is it's the only treatment which produces long-term multiple complete remissions. Another group of prolonged stable disease patients exist. At the time these studies were done, stable disease wasn't really considered an endpoint. We now know in treating kidney cancer by studies done with the targeted therapies that stable disease is in fact a therapeutic benefit. And we can stabilize people, again, for months to years by giving immunotherapy as well as the targeted therapies. It's a small number, it's highly selective, and it's complex. And so for me, it's what we had for 20 years. We're still going to use it. We're going to explore new ways of using it. But it tells us that immune therapy really is something we should capitalize on in taking care of patients with kidney cancer. Now, Dr. Wong mentioned this briefly, but there are some new vaccine strategies. Vaccines for cancer in general have had a lot of bumps along the road, and we've been doing it for more than 20 years. But the key is, what is the antigen that the immune system will recognize? Like when we get a vaccination for measles or mumps, we know the virus, so we can target those proteins. Here, it's harder to know, and so one of the thoughts and one of the ideas is this Argos trial that Dr. Wong mentioned, which is personalized immunotherapy, and it's kind of a neat idea. Basically, what dendritic cells are, these are part of the immune system. They're called antigen recognition cells. They take the tumor cell and they chew it up and they spit out proteins that are characteristic of the tumor for your T cells to recognize. So it's a cell-cell interaction. But by using part of the tumor, see it says, I can't even say it, electroporated patient-amplified tumor RNA, what you do is you take a piece of the tumor and you mix it up with the dendritic cells, and then the dendritic cells say, this is the tumor antigen you go after, and the T cells are going to go after it. And so that's a concept of where your tumor can be the protein that your immune system will chase. So it's a clinical trial. We don't know yet whether this is the answer, but clinical trials are how we've made progress. So that's why with the Kidney Cancer Association, we're always talking about clinical trials. It's an expansion of the T cells, your own T cells, and these RNA-loaded dendritic cells. It is designed to induce an immune response. It's an international study, and it's basically after the kidney's removed, then this vaccine is produced. So it's, again, as he mentioned, it's sunitinib alone versus sunitinib plus the vaccine. So we shall see, okay, but it's stay tuned. Preliminary data showed that there were durable immune responses achieved with significant increases in these T cells. Now, whether that's going to lead to anti-tumor effect, we're going to find out. The other thing that's important at the bottom is that you need to know about when you're talking about immunotherapy is it's a decrease in regulatory T cells and myeloid-derived suppressor cells. Keep in mind that the immune system has to have checks and balances. One of the side effects of some of these immune-active therapies, including some of the targeted therapies, is that we can induce autoimmunity, meaning immunity to normal tissues. And so in normal circumstances, your body's immune system will go after something that's bad, like say you got a bite or something and you get a swollen lymph node and there's an immune response, and then it calms down and it goes away. And that's because other cells are saying, okay, you've done your job, turn off. So we have a push-pull in the immune system. And these regulatory cells and these MDSCs can actually turn off an immune effect. And that's one of the issues with, say, tumor-infiltrating lymphocytes, is a lot of them are regulatory, and they're actually turning off the immune response. So it's important to, so that's something else that we're fiddling around with, and certainly the laboratory scientists that are immunologists are trying to figure out how to enhance the killer T cells and not activate the regulatory T cells. So Dr. Alter brought up the story of the checkpoint inhibitors. Anti-CTLA-4 is Yervoy, is one of them, and it is approved for melanoma. It, again, takes the brake off the immune system. It's kind of a global activation. There is activity in renal cell, but there was a lot of GI toxicity during the initial studies in renal cell, so kind of people have shied away from it. Part of that was just an inexperience with this drug. You know, with every new drug we have, there's what's called a learning curve. We have to learn how to recognize the problems. And I think this may have been just, you know, one of the early kind of casualties of learning how to use Yervoy. On the other hand, the anti-PD-1 and anti-PD-L1 work at a little bit further down in the immune system, closer to the tumor. They're actually, the tumor can inactivate the T cells. And so by putting that antibody in between the T cell and the tumor, then the T cell can go after the tumor again. The tumor actually can block the cytotoxic T cells. And so the PD-1 doesn't have as much of a generalized autoimmune activation as does the anti-CTLA-4. So in, you know, as I said in the previous slide, an older population with other medical issues, we're thinking, not proven, but we're thinking that the anti-PD-1 version of checkpoint inhibitor may be more manageable and certainly will be a candidate for combination studies. So, as I said, they're still in clinical trials. From the phase 1 studies, there are responders and even long-term responders published in melanoma. We don't know yet the story in renal cell. And the toxicity is autoimmunity because we're activating the immune system. So the stuff that's been published so far, and it's still early on, these were broad phase 1 and 2s. They took patients with a lot of different cancer types, including kidney cancer. You can see some of the toxicities in the second bullet. They're immune-related. They're called rash, diarrhea, pneumonitis, and then colitis, hepatitis. Hypophysitis means they knock out the pituitary gland, thyroiditis. So there are immune toxicities. But as you can see, with renal cell, a small number of patients were treated. Response rate about 27%, and stable disease greater than 24 weeks, 27%. So a respectable outcome. Obviously, these are being pushed right now in melanoma. They've been approved for melanoma. But they're also, I think the fact that they're looking at diseases such as non-small cell lung cancer and seeing something that looks fairly respectable should also be kind of hopeful for the non-clear cell renal cell patients because, non-small cell lung cancer is not clear cell. So it may well be that this type of a broader immune activation may have applications to other types of kidney cancer. We just don't know yet. And this is the other phase 1/2 study that was published, and again, there were responses in almost all of the tumor types. And this is the renal cell. It was a little bit less than the previous study, but again, a lot of stable disease. And when I always say in renal cells, stable is good. It means there's an activity there. So we shall see. But, once a drug is out there, we learn a lot more about it than we ever learned from the clinical trials. So this is just to say that anti-PD-1 antibody produces objective responses in patients. Durable responses greater than a year were observed in 20 of 31 patients with melanoma. We'll see. And David McDermott has published on the renal cell cohort, again, 34 for safety, 33 for clinical activity, and 9 of 34 had greater than or equal to a partial response, 1 complete response. Durable some out to more than 2 years. So ongoing. Stay tuned. I'm not going to go with that. So what are the future directions? Well, the first question is, can we step on the gas and the brake at the same time? And so there are clinical trials now in development to combine anti-PD-1 with interleukin-2. And what the game plan is to give a standard dose of PD-1 and then slowly dose escalate the IL-2. And one is being done in both renal and melanoma by the cytokine working group, and the group at Rutgers is opening up a study in melanoma. And I think everybody's been waiting for these studies to start to happen, and finally, I guess because one of the drugs is now, well, both of them now are commercially available, we can get our hands on those drugs. So this is going to be very important, and it may be a big step forward in immune therapy. We shall see. So, and ongoing investigations, I just want to mention something that I'm working on, which is the association of renal cell cancer and hematologic malignancy in the same patient or in families. We don't know why, but it's more than would be expected from the SEER database. So it could be immunologic, it could be genetic, or it could be environmental. And we are collaborating with some molecular biology laboratories to at least look at the genetic component, and then we'll see where things go. So thank you very much for your attention and the opportunity to speak.
Good morning, everybody. So I'm going to talk about immunotherapy for the treatment of metastatic kidney cancer today. So as many of you probably know, immunotherapy was once regarded as an ancient therapy of almost the Jurassic period. However, recent progress in research has really revived the dinosaur immunotherapy for treatment of cancer. So I'm going to spend some time with you today to review the history of immunotherapy. And more importantly, I'm going to share with you some exciting data in the field of immunotherapy, especially for the treatment of kidney cancer. So why are we interested in immunotherapy? Well, we know immune system can kill cancer cells. However, the immune system kills cancer cells in a different way from the traditional chemotherapy and the target agents. First of all, it's adaptable. So when the cancer cell changes, the immune system can also change accordingly. Second, it's specific. So the immune system will target the cancer cell directly and spare the normal healthy cells. Third, it has memory. So as you know, cancer cells can hibernate in your body for many years. So even after 10 years of hibernation, when these cancer cells wake up, the immune system also wake up, try to catch these cancer cells. And there are quite a few reasons for us to develop immunotherapy as treatment for cancer patients. First, we know tumors with more infiltrating lymphocytes actually are associated with good prognosis. Second, we know that mutations during cancer development and progression actually can create these new antigens. And these new tumor antigens can be recognized by the T cells and the immune system. Through history, many strategies have been used for immunotherapy. The earliest strategy was to just use bacteria stimulants to stimulate the immune system, which I will talk about more in a moment. The second one was to use cytokines to energize the immune system to kill cancer cells. People also tried to use vaccines and adopt T-cell therapy. So these two methods are not used for kidney cancer. Therefore, I'm not going to talk about them today. But I will spend the majority of time to talk about immune checkpoint blockade therapy, which really has shown some great potential for kidney cancer. So the bacterial products were first used by William Coley to treat cancer, known as Coley's toxin. So Dr. Coley actually cured the first patient with these bacterial products over 120 years ago. And later on, it was found it was interferon alpha, actually, that was stimulated by this Coley's toxin to kill cancer cells. And he treated quite a variety of tumors. And the tumors he treated the most was soft tissue sarcoma. And as you can tell from this data, he treated more than 100 patients. And over 50% of these patients can live more than 5 years. For kidney cancer, he only treated 6 patients at that time. 3 patients didn't have response. The other 3, which is 50%, actually lived more than 5 years. So even by today's standard, Dr. Coley was quite successful. So why Coley's toxin was not approved for treatment of kidney cancer? That's because it's too toxic. So after patients receive this Coley's toxin, they have shaking, chills, they really become very sick. Therefore, FDA actually didn't approve Coley's toxin for treatment of kidney cancer. And then almost 100 years later, people found IL-2 actually has activities against kidney cancer. That's because IL-2 can stimulate T cells and the natural killer cells to kill tumor cells. Other than IL-2, interferon alpha and interferon beta were also shown to have modest activities against kidney cancer, as Dr. Tenier just talked about a moment ago. So this is the high-dose IL-2 data on a total of 255 patients. 37 patients actually had response. So of these 37 patients, 17 patients had a complete response. But please note, of these 17 patients, 80% of them lived up to 10 years and beyond. And for those patients who had only partial response, and there are 20 of them in total. So only 30%, actually only about 25% of them lived to up to almost 10 years. So really, IO2 therapy offers the long-term survival potential for patients. However, the response rate is pretty low. So the complete response rate is lower than 7%. The partial response rate is lower than 15%. And it is, again, a very toxic therapy. So many patients who receive high-dose IO2 have to be monitored in the ICU setting. Therefore, IO2 therapy is really limited to the younger and healthier patients. And then Dr. Tanier already showed you this piece of data by combining interferon alpha and bevacizumab. As you can tell from the table here, the combination therapy actually can improve the progression survival of patients, which means it delays the tumor recurrence or progression by about 3 months. And because of this, bevacizumab plus interferon alpha, along with high-dose IO2, were approved by the FDA for the treatment of metastatic kidney cancer, along with many target agents that were discussed by Dr. Tanier just a moment ago. So this is basically the timeline of the treatment options for renal cell carcinoma. So in 1980s, IO2 and interferon alpha were first shown to have activities against kidney cancer. And then 10 years later, high-dose IO2 was approved by the FDA. And over the past 10 years, 7 to 10 years also, there is just an explosion of these target agents, which are really great options for treatment of metastatic kidney cancer. And because of these agents, people's life, people with metastatic disease, the average survival has increased from about a year to more than 2 years. And this is the combination of bevacizumab plus interferon alpha. However, despite the recent success of these target agents, we all know that many of these patients will develop resistance to the treatment just within months. So after that, we have to switch to another agent. So the question is, do we have any better therapy with long-term survival potential? And then that's when the immune checkpoint therapy enters the picture. So what is immune checkpoint therapy? I have to talk about immunology just a little bit here. So in order for the T cells to be activated for tumor cell killing, it needs 2 sets of signals between the antigen-presenting cells and the T cells. The first set of signal is delivered by the interaction between this T cell receptor and the tumor antigens bound to an MHC molecule. The second set of signal is provided by CD28 binding to B7. So in a way, this is like driving a car, okay? So the first set of signal is like the ignition system. So you need to turn on the car. And the second set of signal is like the gas pedal. After turning on the car, you need the gas pedal to speed up the car so that you can kill tumor cells. However, we all know if we only have the ignition system and the gas pedal, and you drive a car, sooner or later you are going to run into problem because you are going to speed up and you cannot slow down and you're going to crash and burn. So the immune system is actually very smart. It actually has its own braking system. One of these brakes is called CTLA4. The other one is called PD1. They are responsible for slowing down the immune system after it's activated. However, the side effect of these brakes would be decreased tumor cell killing. So James Anderson, who is the director of the immunotherapy platform at MD Anderson, found that if you use an antibody to block this inhibitor, to block these brakes, to take the brakes off these T cells, you actually can allow the T cells to stay activated for augmented tumor cell killing. And that's how the first drug, ipilimumab, is developed. So ipilimumab is basically a monoclonal antibody against that molecule named CTLA4 here. It was first tested in advanced melanoma patients, who on average has a survival of about a year, just like metastatic kidney cancer. However, as you can see from the data here, after these patients receive ipilimumab, over 20% of them can live up to almost 5 years, and that is quite significant. So what if you put ipilimumab and the other antibody that breaks anti-PD-1, which is named nivolumab? So again, this is in melanoma patients, as you can tell from the data here. So on average, please remember, on average, these patients live for about 12 months. But here at 1 year, 82% of the patients are still alive. By 3 years, over 70% of the patients are still alive. And that is very, very dramatic. And that's why immune checkpoint therapy was offered Breakthrough of the Year honor by the Science magazine. So what about these therapies in kidney cancer? So based upon these phase 2 trials at lower concentration, ipilimumab didn't have very much effect in kidney cancer. However, at the normal concentration that we use for the treatment of metastatic melanoma patients, 5 out of 40 patients actually had partial response. For nivolumab, the result is even better. So at 6 months, about 24% of the patients actually showed partial response, and 24% of patients actually showed stable disease. So overall, almost 50% of patients actually benefited from nivolumab at a very low concentration of 1 milligram per kg. At a higher concentration, the response rate increased to over 30%, and the stable disease rate increased to over 30%. So overall, more than 60% patients actually benefit from nivolumab. So what about putting those two drugs together? We don't have data yet. Recently, we just designed a clinical trial to combine either nivolumab with ipilimumab or bevacizumab, which is a standard treatment for kidney cancer as discussed by Dr. Tanier. So we designed this 3-arm trial. In the first arm, patients will receive nivolumab. In the second arm, patients will receive nivolumab plus bevacizumab. And in the third arm, patients will receive the combination of nivolumab plus ipilimumab. After 6 weeks of treatment, they will have surgery to remove the kidney with disease. And if they have either stable disease or response before the surgery, they will continue on nivolumab. So this trial actually has quite a few advantages. First of all these patients will receive at least double therapy or even triple therapy. So patients in arm 1 will get nivolumab plus surgery, which is a standard treatment for metastatic disease with the intact kidney tumor. So patients in arm B and arm 3 will receive triple therapy. Second of all, this design will allow us to collect blood samples before and after each trial, and also collect tumor samples before and after each treatment. This way, we can study the biology of these drugs directly in human body, and hopefully we can identify biomarkers to improve these therapies, and also identify new targets for further therapy. So with these target agents, we know we can prolong survival a little bit. And with immunotherapy, we can see this tail-end effect, which is long-term survival. And by combining these immunotherapy agents with each other, and also with the traditional target agents, we hope we can not only prolong survival, but also increase the response rates in patients with metastatic kidney cancer. With that, I would like to thank the collaborators from the immunotherapy platform, from pathology, GU oncology, and also urology. But above all, I would like to thank our patients and their families for your support with our research and clinical trials. And I hope we can continue to work together to rewrite cancer history. Thank you very much for your attention.
So to complete this morning's session, there's a little bit of a change in the schedule. Our next talk is actually appropriate, going to be our vaccines and novel immunotherapies promising in the treatment of kidney cancer. And we're joined by Samantha Paul, who's the director of the kidney cancer program at City of Hope Comprehensive Cancer Center. Thanks for coming, Monty.
Thanks so much, Dr. Figlin. My name is Monty Powell. I'm over at City of Hope. And before we begin, I really just wanted to give a huge thank you to Dr. Figlin and Nancy and the KCA for organizing this conference every year. Many of us in the audience know Dr. Figlin as a superb clinician and someone who's really brought a number of new therapies in kidney cancer to the helm. But what we don't often see in our day-to-day interactions with him in the clinic is that he's a really outstanding teacher. And I give him full acknowledgement for teaching me everything I know about the disease. And hopefully that's a good thing. So I'm actually going to answer the title of my talk in just one slide. So are new immune therapies and vaccines promising? I think the answer to that is certainly yes. But what I'll suggest, it's still pretty early. So we're going to go over a couple of clinical trials that are ongoing right now, some that are evolving here at Cedars-Sinai, some at City of Hope, that may ultimately bring an answer to this question more definitively. Dr. Hoffman gave a great outline of the developments that have been made in kidney cancer over the past several decades. We saw the approval of interleukin-2 back in 1992, but it's really been over the past decade that we've really seen a number of targeted therapies evolve. And as Dr. Hoffman outlined, these are all targeting blood vessel growth and formation by and large in kidney cancer. So I just wanted to very quickly give you a glimpse of the older immunotherapy data. These are the immune-based treatments that were used in years past. Dr. Hoffman had alluded to interferon therapy, and I agree with him completely. What we saw with this, I'm just going to highlight a couple of key points that he's mentioned, are that response rates are very low. So we see a small proportion of patients who actually generate a response to interferon therapy. I'll also point out that we actually don't see many durable responses with interferon. This is in contrast to interleukin-2, which we'll talk about in just a moment. I'll also propose that the median survival, although this was sort of a benchmark for us a decade ago, was unfortunately not very good with interferon therapy. So if a patient was sitting in my clinic a decade ago and asked me, Doc, what are my odds of making it past a year with a diagnosis of kidney cancer, I'd really have to quote to them some pretty disconcerting data in association with this drug. And it really didn't do a whole lot to delay cancer growth. We saw that in terms of a delay in cancer growth, interferon slowed down tumors for a median of around 5 months or so. And this was actually the benchmark, as I mentioned before, that we've really improved upon with some of the targeted therapies that we had at our disposal now. So you just heard a moment ago from Dr. Hoffman about a clinical trial that compared sunitinib to interferon therapy. That was a trial that Dr. Figlin actually led. And sunitinib really trounced interferon therapy in the context of that clinical trial, and it's really moved ahead in terms of our pipeline of agents. Well, what about interleukin-2? Here I'll also concede that response rates in broad terms are very low. In terms of the response rate that Dr. Hoffman referred to, around 15%, we see that that's typically the case in association with this drug. The rates of durable response, so these are the really meaningful responses that we see in association with the drug, these are even lower. So the way that I typically summarize the data for interleukin-2 is that if I've got a room full of 100 patients, so we have 5 rows of 20 patients each here, we're going to have 5 to 7 patients in that room who have a really good long-term durable response. And in fact, we've got some of those patients sitting here in this room today, which is really outstanding. But the unfortunate reality of interleukin-2 is we are going to have some patients who die as a consequence of the treatment, and we're also going to have a number of patients who really don't respond to therapy. If I had to summarize the targeted therapies in one slide, this is perhaps how I do it, and this is generally how I would walk my patients through this in the clinic. If we have that same 100 patients sitting within the room, we're going to have 40 patients within that room have a response to therapy. We're going to have another 40 patients in that room have stabilization of their disease with therapy, but we're going to have 20 patients in that room whose disease unfortunately continues to grow. So this might be an overly simplistic way of looking at this particular problem, but what I would also posit is that with the targeted therapies, we don't have those long-term durable responses by and large. We're starting to see those evolve, but maybe the proportion isn't quite as high as with interleukin-2 therapy. How do we predict who benefits from interleukin-2? My personal contention is that with the older immunotherapies, we really don't have a great way of identifying that. And this is going to be a distinction with some of the newer targeted immunotherapies that we have at our disposal. There was a trial called the SELECT study. Again, Dr. Figlin was one of the leaders in this trial. They looked at a total of 126 patients, so this was a relatively large effort. And the main aim of this study was to determine whether or not there was anything within a tumor sample or amongst patient characteristics that might predict clinical outcomes. So we could take that big room of patients and really weed out those individuals who might derive particular benefit from interleukin-2 therapy. But I would suggest that this study was ultimately negative from that standpoint, and still in 2014, we can't predict who these 5 to 7 individuals in the room are going to be. So what's the future of immune-based therapies? And this is really going to be the focus of my talk today. We're going to divide this into 2 different subjects. The first is the use of programmed death 1 inhibitors, or PD-1 inhibitors. And this is a very ominous name, but this drug functionally actually works in a way that's very similar to interleukin-2. We're really trying to stimulate the body's own response against the cancer. Vaccine therapies fall into 2 categories, and I'll talk about this. The first is autologous vaccines, which are actually derived from a patient's own tumor. And the second category is termed multipeptide vaccines. These are vaccines that can actually sit on the shelf in a clinic that we can deliver without actually having the benefit of the patient's tumor to derive the vaccine itself. And again, these agents all harness one particular weapon against cancer, and it's something you're probably going to hear more and more about as cancer therapies evolve, and that's something called the T cell. So this is the T cell. It's one of the body's immune cells, and it really is responsible for taking on many of the cancer cells that invade patients with cancer. The T cell interacts with something called the antigen-presenting cell, and I really do think that understanding this is essential to figuring out how these PD-1 inhibitors work. So when cancer enters into one's body, basically portions of that cancer are shed out into the bloodstream. Those portions of the cancer are displayed on the surface, this green surface here, of the antigen-presenting cell. And when the T cell comes into contact with it, at this particular interface, we actually have stimulation of the T cell, and it begins to respond against the body's tumor. This is perhaps a simplified way to look at that interaction, and it explains how some of these PD-1 inhibitors work. This is, again, that interface between the T cell and the antigen-presenting cell. We have the factors on this side that are actually stimulating that T cell to generate a response against the cancer. But, you know, as with everything in the body, there are checks and balances. So on the left-hand side of this diagram, what you see are those balances. Essentially, these are proteins that will interact with one another and actually shut down the T cell's response. So what we really want to do with these novel immune-based therapies is in a very targeted fashion shut down this interaction. And that's precisely what we do with the PD-1 inhibitors, or the programmed death 1 inhibitors. We actually interfere with the interaction between PD-1, which is a protein that sits on the surface of the T cell, and PD-L1, which sits on the surface of the antigen-presenting cell. So as you look across the internet, clinicaltrials.gov, other references, you're going to hear about a whole host of therapies that actually target PD-1 and PD-L1, and this is the mechanism on which they're all predicated. And I'm happy to answer questions about that as soon as this presentation is done. So how well do these drugs work? Well, I'm actually going to focus on data for the one agent that perhaps has been tested to the greatest extent in kidney cancer so far, and that's a drug called nivolumab. So this is a PD-1 inhibitor, and just to rehash the mechanism of this agent, basically it's blocking that interaction between PD-1 and PD-L1 by binding to this very specific site at the surface of the T cell. To date, there was a study performed, a big phase 1 trial that didn't just include kidney cancer. It also included lung cancer, colorectal cancer, pancreatic cancer, and a whole host of other types. The study actually did include a total of 34 patients with kidney cancer. And what they found, pretty remarkably, is that at the 2-year mark, about half of patients were still alive. Now, that in and of itself might not seem remarkable, but when you consider how many treatments these patients had had prior to entry in the trial, I think it's truly fascinating. So about half of patients in this study had gotten 3 or more prior treatments. What I think is also pretty interesting is we're seeing that same phenomenon of the durable response evolve. So we see that amongst these patients, we had 30% who generated a response, many where the response is very long-term. You see here 96 weeks on the x-axis here, so fairly long responses. One thing that also is pretty bewildering to us in the clinical community is that we're actually seeing responses that last well beyond the administration of the drug. So what you see here in the white bubbles is actually when the response was generated with these agents in particular patients. Each one of these lines represents a specific patient. And what you see here is the time for which the response evolves and the treatment is continued. But in the red bars, we're actually seeing responses that last well beyond the treatment is discontinued. So it's almost like being on drug X and having that response last well beyond drug X. And we're seeing that phenomenon more and more with this class of agents. It's truly something to look forward to. Now, I do want to insert one word of caution here. What I've presented to you is data in 34 patients. This was in the confines of a phase 1 trial, and those of you in the audience that are familiar with clinical trials know that we go through a whole battery of different criteria to facilitate enrollment. By virtue of that, there are sometimes big selection biases in who we pick and don't pick for these clinical trials. So I would suggest that these 34 patients, who again had been through a mean of 3 or more prior therapies, are probably fairly healthy folks, and they might not represent the norm for kidney cancer. So I'd urge us to interpret this data with caution, but I would say cautious enthusiasm perhaps. I'll point out that I've been involved with 2 other studies. This is a phase 1 trial of a drug called XL184. It's now known as cabozantinib. And this was a pretty impressive drug in the phase 1 setting. We treated a total of 25 patients, many of whom were at City of Hope. And this was also a pretty healthy group of folks. They'd already gotten 2 prior therapies, but nonetheless met all criteria for this trial. And their tumor delay and growth was actually over a year. So in the context of the treatments that we have at our disposal now, that's a pretty impressive bar. I also led a study, and I put this up here really to, again, exert a word of caution in interpreting the PD-1 data of a drug called pazopanib. And Dr. Hoffman alluded to this drug. This is one of the standard of care agents. It's FDA approved. We could deliver it in the first line setting. When I looked at this drug after patients had gotten 2 prior lines of treatment, we really saw a phenomenal delay in cancer growth of 17 months. And in fact, the majority of patients on this trial are actually still alive today, 4 years after the trial was conceived. So whenever we look at clinical trial data, it's important to really take a close look at the patients that we're enrolling in those efforts. So how are PD-1 inhibitors going to move forward? This is a trial that Dr. Figlin is actually running here at Cedars-Sinai. This is a trial that takes patients with metastatic kidney cancer. They may have received 1 or 2 prior anti-angiogenic therapies. Again, these are drugs like sunitinib or sorafenib or what have you. And very appropriately, I think it randomizes them to everolimus, which is a drug that we might typically prescribe in that setting, or you might actually get the PD-1 inhibitor. And this trial is still ongoing. I think enrollment is close to completion right now, but it'll enroll a total of 822 patients. And this trial might actually be the one to get PD-1 inhibitors on the market in renal cell carcinoma. So this is definitely a trial whose results I think we're going to look forward to. I also wanted to highlight a different trial that we're running at City of Hope. This is an exciting one. This actually takes 3 different study arms, and I'm going to spend a little bit of time going through this. We heard from Dr. Hoffman earlier and from Dr. Kim that there are several promising agents already in our pipeline, like sunitinib and pazopanib. What if we were to actually combine those with a PD-1 inhibitor? That's exactly what we're exploring at City of Hope. So we've got the combination of sunitinib and nivolumab, that same PD-1 inhibitor I referred to in the previous slide. We're also looking at pazopanib with nivolumab. And in what I think is maybe the most intriguing arm of the study, I certainly can't say the best, but perhaps most intriguing, is the combination of ipilimumab and nivolumab. Ipilimumab is an immune-based therapy, and if I could go back a couple of slides to the surface of the cancer cell, ipilimumab actually blocks a very different interaction at that T cell and antigen presenting cell interface, whereas nivolumab blocks a different set of proteins. So in this clinical trial, what we're actually looking at is this particular combination of drugs. So 2 immune-based therapies given to the patient at once. And I think hopefully that'll shed some light on optimal treatment strategies using these immune-based agents. I'm just going to very quickly allude to a trial that's opening shortly at City of Hope. This is another exciting one that's actually going to potentially move PD-1 inhibition into the frontline setting. And this is for patients who don't have metastatic disease at the moment. What it will do is it will randomize them, once they do develop metastatic disease, to a PD-L1 inhibitor that's being made by the company Genentech. They could receive that drug alone or in combination with bevacizumab, which Dr. Hoffman alluded to correctly as being one of the standards of care for treating kidney cancer, or sunitinib, again, a very valid frontline option. So this trial will be opening up within the next 3 months at City of Hope. We're very excited to have it on board, and hopefully it's one that would move PD-1 inhibition to the forefront of therapy of renal cell carcinoma. I'm going to also talk about vaccine therapy, and again, we're so fortunate to have Dr. Figlin here, who's been really leading the charge in terms of vaccine therapy development for kidney cancer. What I've alluded to on this slide, and this is a relatively busy one, is that in the domain of vaccine therapy for cancer, we've got Provenge, which is approved right now for prostate cancer, but we've got a whole host of other agents, Simuvax, DCVax, AGS-003, basically alphabet soup of vaccine therapies that are currently in the pipeline for other cancers, including kidney cancer. And my goal today is to very quickly outline two of the agents, two of the vaccine therapies that are currently in the pipeline for kidney cancer. I'm going to first talk about an off-the-shelf vaccine. So recall earlier I talked about two different vaccine types, one that we could administer to the patient without any preparation, something that's essentially standardized across all patients. And then we've got the autologous vaccine, which is really patient-specific. It's derived from the patient's own tumor. This is actually one of those off-the-shelf vaccines called IMA901. It was actually generated through a very complex algorithm, and rather than going through this, what I'll suggest is that they really went through an extensive process to dichotomize, to really figure out differences between normal cells and tumor cells. And what they've identified are key protein-based differences, and it's really those proteins that they're targeting through this vaccine. There is a trial that's completed enrollment right now that takes patients with newly diagnosed kidney cancer and randomizes them to get this vaccine with sunitinib or sunitinib alone. And again, this is the sort of trial that really may push vaccine therapy to the forefront of treatment in kidney cancer. This is a trial that I give Dr. Figlin full credit for. He's been championing this for some time now, and it's actually in the phase 3 mode of testing, which is incredibly exciting. This is looking at a compound called AGS-003. And this is really where we hope most treatments in cancer will go. This is a highly personalized approach. Basically, when one is diagnosed with kidney cancer, they have their tumor removed. The DNA and RNA-based elements, the genetic information within their tumors, are actually siphoned out, essentially, and a vaccine is developed by taking that genetic information and actually more or less plugging it in or forcing it into immune-based cells. So this, again, is a very highly personalized approach. And at City of Hope, when Dr. Figlin actually headed our department, he ran this trial looking at 21 patients with metastatic kidney cancer. These patients all received sunitinib in combination with the vaccine. And one thing that I'll point out is that these patients, by virtue of having a tumor in their kidney and disease within their lungs and bones and other sites, were what you might call intermediate or poor risk from the get-go. Dr. Hoffman had alluded to some of these risk categories. So the expectations for survival from the get-go were not great. But essentially what Dr. Figlin's data shows with a combination of the AGS vaccine in combination with sunitinib is that patients with poor risk and intermediate risk disease were living 5 times as long perhaps as we might expect. So that's an oversimplification of this diagram, but I do think that that data was incredibly encouraging. And so what Dr. Figlin is leading right now in combination with a surgical colleague from MD Anderson is a phase 3 clinical trial. And in that study, patients who have, again, disease that's present in the kidney and at metastatic sites, randomized to receive either sunitinib or a standard of care treatment like pazopanib, and they'll receive that in combination with the personalized vaccine therapy. So trials like this, I think, may potentially, again, move vaccines to the forefront of treatment in kidney cancer. So I would suggest that the response to targeted therapies isn't bad. Recall that, if we have these 100 folks in a room, we're going to have the 40 who have a response and shrinkage of their disease, the 40 who have stabilization, and the 20 who progress. But what we really want to do is harness that benefit that we had in the interleukin-2 immunotherapy era and generate some durable responses. But one thing to keep in mind is that these targeted agents are actually immune-based drugs as well. Drugs like sunitinib may potentially augment the immune response, and drugs like sorafenib may actually have the opposite effect and inhibit the immune-based response. So as many of these trials manifest, you're going to see combinations of sunitinib and pazopanib with the PD-1 inhibitors, with vaccine therapies, and it's going to be really interesting and important for us to figure out how these treatments all interplay. So again, hopefully with targeted therapy and immune therapy in combination, we'll turn everybody in this room into a durable long-term responder, and that's certainly what I'm hoping for. In the clinic, a lot of folks ask me, and I'm going to diverge here for just one moment and ask, what would you do? So this is 2014. We've got 7 FDA-approved agents beyond interleukin-2 at our disposal for kidney cancer. When folks ask me about my approach, I typically divide our therapies for kidney cancer into 2 buckets. We've got our VEGF-directed bucket with sorafenib, sunitinib, bevacizumab, axitinib, and pazopanib. These are drugs that many of you in the room may have been exposed to. We've got our mTOR inhibitors, everolimus and temsirolimus. What I really think we've got to do is get outside of these buckets, and I always recommend participation in clinical trials. We have a whole variety of clinical trials available at City of Hope. There are different and important trials available here at Cedars-Sinai, others that Dr. Quinn, for instance, may offer at USC. And this is a very busy diagram, but it shows you the algorithm that I tend to follow when I treat kidney cancer therapy. We have trials that are intended for patients with localized disease. We have trials for patients who, for instance, have one metastasis in the lung that's resected. The goals of those trials is to prevent the risk of recurrence. And we have trials for patients who have metastatic disease who have had 1 prior line of therapy, or 2 prior lines of therapy, or 3 prior lines of therapy. So by having these various clinical trials at our disposal, I think we really can potentially stay outside of these 2 buckets and offer our patients with kidney cancer a maximal number of diverse therapeutic options. I'm just going to spend one moment going over the list of trials that we have available at City of Hope right now, just to give you a sense of what those outside of the bucket options are. I've already highlighted this particular trial, which I would offer to patients who have newly diagnosed metastatic kidney cancer. This again takes that PD-L1 inhibitor, the novel immunotherapy, and randomizes patients to that or sunitinib or a combination of drugs. In the second line setting, we're offering a clinical trial looking at that drug XL184, which I'd mentioned had very compelling phase 1 data. In the third line setting, we're offering 2 clinical trials, one looking at a stem cell antibody, which I think is really promising, another looking at a novel immunotherapy called sonapsizumab. This is being developed by a small company down in San Diego. If a patient has had 3 prior lines of therapy, we've got combinations of the PD-1 inhibitor with VEGF-directed treatment, as I alluded to. And if patients have exhausted all of these clinical trials, we yet still have other studies. This is involving a drug that parallels the VEGF signaling axis, but is nonetheless different, called AMG 386. So again, by staying outside of these buckets, by maximizing the number of treatment options that we have available for kidney cancer, I really think we can improve clinical outcomes. Thank you.
We have a few moments for questions, otherwise we'll go to lunch. Any questions? I'm hungry too. Thank you, Monty. So we will have lunch, and you'll be able to bring your... Where? Oh, microphone, please. Thank you.
It sounded like all of the clinical trials are geared to clear cell. Is that true?
Yeah, so a fantastic point. I don't know if we could launch the slides again for just one moment there. If we can't, it's okay. But what I'll point out is that we actually do have a couple of clinical trials that are available for non-clear cell disease as well. Let me actually fast forward to this clinical trial algorithm here. I'm really glad you brought that up, because non-clear cell disease is critically important. Non-clear cell, by the way, represents about 20% of kidney cancers. We have papillary kidney cancer, which is about 10 to 15% of all kidney cancer diagnoses, and chromophobe type kidney cancer, which is about 5%. These are not insignificant proportions of the population, so we've definitely got to address them. So I'm going to jump forward to that clinical trial algorithm that I showed you earlier. And in this clinical trial algorithm, one thing that I'll point out is that if you have localized kidney cancer that's been resected and we want to employ a preventative therapy, we do have a trial called the Everest study. And it's hard to see over here, but I'm happy to go through the particulars with you. And this randomizes patients to everolimus in the setting of that particular stage of disease. In the metastatic setting, several of the trials, and I've really been working towards this over the past couple of years, to have trials in my portfolio that do actually address the non-clear cell population. We have this trial of a stem cell antibody called TRC105 that's led by Dr. Quinn's group at USC, and they actually do permit non-clear cell histologies, including papillary and chromophobe subtypes. Within the Southwest Oncology group, I'm actually taking the lead on a clinical trial that's going to be looking at about 5 to 6 different MET inhibitors. We think that MET is a protein that drives a lot of papillary kidney cancer. So that trial may evolve over the next 2 to 3 years or so, and that may be an efficient way of determining what the optimal therapies are for that particular disease. So I'm so glad you brought that up, and I do think non-clear cell disease is critical, and I think that the bottom line is we're definitely focused on that in our clinical research efforts. Thanks.